So originally when I was hired for this project, it was to help close the carbon and nitrogen budget. And my role in doing that was to provide data concerning trace gases in soil respiration, carbon dioxide, methane, and nitrous oxide, along with carbon in the soil, dissolved organic carbon, and nitrogen in the soil, dissolved organic nitrogen, as well as ammonium and nitrate. With those analyses, I also look at soil water content to help analyze the other indicators that I talked about. Because this project was growing, I was also able to bring in the component of soil microbiology. So I look at the soil community given different management practices, and so I look at bacteria, fungi, and actinomycetes in annual and perennial systems among the southern plains. So generally people think about things they can see or living creatures in the sense of animals or humans that respire. The soil respires just like we do, given microbes or the fauna, the roots that are in that system, and the insects that are in that system. The other way that the soil respires is through decomposition of soil organic matter, which is mediated by microbes. And so they can respire CO2 just like you and I in an aerobic situation, but in an anaerobic situation they can respire a multitude of different products, those of which that I am focused on are methane and nitrous oxide. So in soils we have two key players that play in the world of methane. We have methanogens, who are the methane producers, and we have methanotrophs, who are the methane consumers. And generally methanogens are associated with rice paddy production or ponded soil. We don't have that phenomenon here in the southern plains, and when we do, it's rare. And we generally deal with methanotrophs who consume atmospheric methane as their carbon source, and they actually respire CO2 and do so with arid soils that are highly aerated, because it can be an aerobic or an anaerobic process, but generally aerobic in this region. And they respire CO2 and take up that methane out of the atmosphere. And they essentially cause that soil to be a sink for methane, and that could be very, very beneficial when we start talking about global warming potentials of an ecosystem or a management practice. Generally, microbes, good rule of thumb is the more the system is disturbed, whether that be through tillage or chemical treatment, animal grazing or whatnot, there are less microbes that are active. However, in native systems or grassland systems, perennial systems that have reduced management, there's generally a higher affinity for consumption opposed to flux. So what I presented today was essentially that native systems, native prairie that are unmanaged, they can still have cattle on them, but they're not fertilized or tilled, have the greatest affinity to consume methane. And as you increase management, whether through no-till and high chemical addition or tillage plus high chemical addition, you see a perfect gradient of the decrease of accumulation or consumption of methane in the soil. Instead, it actually fluxes, particularly in times of high management like summer months after harvest.